And now our next speaker is Prof. Florence Delie from the University of Geneva. From Delie is a researcher associate at the School of Pharmaceutical Sciences in the Laboratory of Pharmaceutical Technology and Biopharmacy in Geneva. She works in the group of Prof. Couvreur at the Paris Sud University and in the Department of Pharmaceutical at Genentech. She has a special interest in the interaction of colloidal carriers with biological barriers, cellular targeting, and in transcellular uptake of polymerical particles. Prof. Delie will be presenting a lecture regarding an in vitro blood-brain barrier model to evaluate colloidal drug delivery system. Welcome.
Thank you very much. Thank you very much for this introduction. Thank you very much also for the organizer for inviting me to this very interesting meeting. And what I found, I learned a lot actually already in 2 days, because in Geneva we are very new in the field of blood-brain barrier crossing, and we know much more about particle making than this, but I'm always excited of knowing and getting more knowledge about how the particle will interact with living bodies. And so when we were approached to go into the blood-brain barrier crossing, we were very excited. And at that point, we said, okay, we do have the technology to make the particles, but we want to make sure that we are able to evaluate how our particle will interact with this type of cells. And as you know, it's quite difficult to have models in vivo. So we looked at the different in vitro blood barrier model to try to evaluate and to see whether it was possible to set up a model in order to screen up the different type of particles that we would be able to make. So I will not go through this slide. We have been many evidences that brain pathologies are very preoccupating at the time, and mainly because the brain is our central control unit of our body and the way that we do interact with all our environment. So that means that every type of pathology that will come to the brain will impair and will lead to very debilitating diseases and disorders. So there is a huge to try to find new compounds and also new way to go into the brain. So this is the reason why the neuropharmaceutical company is one of the largest potential growth sectors in the big pharma. And this is also related to the increase in age-related diseases, related also to the growing older population. And basically the focus for these medicines is both for treating or curing the diseases, but also to have more diagnosis tool to get early diagnosis of this pathology. So to make it very, very short, I think that you all know that the drugs that we are getting are, first of all, going into the vascular compartment, and from there they are distributed in all the organisms, depending on basically their physicochemical properties and the way that they will interact with proteins. The problem with the brain here is that there is a very specific blood-brain barrier that will prevent most of the drugs to get into the brain. There is another blood CSF, cerebrospinal fluid barrier that is also existing. That is not, it's a little leakier than the blood-brain barrier. However, the surface that it will be concerned by this, it's much smaller than the one that is represented by the blood-brain barrier. This is mainly the reason why people are focusing on how we can cross this barrier. So, of course, there is some physiological reasons why this barrier is so tight. It's just to maintain the homeostasis of the brain and to make sure that it will continue to work perfectly. So it's a neuroprotective barrier, and it will maintain a stable environment in the brain vicinity. So if we look at the main actor here in the blood-brain barrier, this is the transmission electron microscopy of the neurovascular unit in the rat. And what you can see nicely here, this is the lumen, so where the blood will go through, and all the flow of the blood will be here in the lumen. And then there will be these endothelial cells that we have talked about, which are very tightly attached together by extracellular and also intracellular molecular complexes. And then we have the basal lamina that is around. And what is very important for the growth of these cells and also for the maintenance of this barrier is the close proximity of other cell types, such as astrocytes that you have here, and also pericytes, even though the clear interference of these cells, the pericytes, hasn't been clearly defined so far. They are very important to be around. The neurons also are very close. So if we look at a schematical representation of this barrier here, so you have the tight junction, so that will link together the cells and that will bring this tightness. There's also efflux protein that will bring drugs or metabolites out of the barrier. And also what is very important is that these cells are polarized. That means that they will not express the same type of markers and transporters on the abdominal side than on the basement membrane or the brain side. So this is very important, and these cells have been quite well characterized so far. And if we are using a model, it's very important that we will be able to find the same type of markers directly in the models. So again, we have endothelial cells in other tissue as well. I just want to point out a couple of differences between endothelial cells and brain endothelial cells. So in the regular tissue, we have fenestration in between those cells, and this does not exist with the brain endothelial cells. They have no pinocytic vesicles, so that means that there is no endocytic activity. They have a lot of mitochondria, so they are very active. They express numerous specific transporters. They do have efflux pumps, and they have, of course, very tight junction. And they also express more cell-cell adherence junction markers. And this is particularly due to the very close interaction they have with the astrocytes or glial food processes, the cells that are surrounding those vessels. So, of course, what we want is to have our drugs getting directly to the brain, but we don't have so many ways so far to do so. So we have, and we have heard yesterday about very invasive method that we can use for those treat these patients. However, this is not always possible, and there are a lot of complications and adverse effects that may be occurring by using this type of way to get drugs into the brain. So we have been also looking as pharmacists to how we can use our knowledge and how we can change the molecules. So this is the chemist part, I would say, where they will change or derivatize the drug directly just by changing its lipophilicity, for instance, or by adding some recognition moiety that will help to cross the barrier. What we have been interested in is to use carrier-based approaches. And mainly, we are using synthetic carriers. Basically, those colloidal carriers that we already have heard about are divided into two groups. I make the difference between liposome and particles. So micelle and liposomes are much more vesicular, flexible structures that are made for the micelles by amphipathic molecules that will lead to have some hydrophobic pocket in which you can have the drug that is dissolved. Liposome, on the contrary, are made of phospholipids that will actually make like a phospholipidic bilayer, and you will have a hydrophilic core, and possibility of having hydrophobic molecules into the membrane. And beside this vesicular system, there are also more solid systems, which are the nanoparticles. Nanoparticles are made out either of polymer, or they can be, as we have seen, with lipids that are getting solid. And we can differentiate different type of particles depending on the way that you are preparing those particles. So you have particles that are made out of a matrix, which are called usually nanospheres. You have also particles that are made of a liquid core that is surrounded by a polymeric wall, and these are capsules. And also we have kind of in-between structures that are made also with double emulsion structures, where you have some domains that are designed here to have drugs with some specificity. So depending on the way that you make your particles, you may end up to have different type of system here. And also you may decorate and you may change the physical-chemical properties of those particles depending on how you make them. So basically the idea of using the colloidal carriers will be to increase the half-life of the molecules into the blood, to have a long circulating formulation, and also to be able to protect this formulation against enzymatic or environment inactivation. Hopefully, this will lead to a better interaction and more penetration into the brain endothelial cells. Hopefully also, and this has been demonstrating by the team of Couvreur with cancer cells, that some of those particles will be able to escape the efflux systems. And also with this type of particles, we will be able to change the contact with the cells, because we will be able to play with different parameters to make the surface properties of these particles quite different. So this is what we have seen. So maybe we can play with the polymer just to change its hydrophilicity or hydrophobicity. We can charge the particles to have them getting through via a certain type of receptors. We can also hydrophilize these particles, and this is very, very important to increase the long time circulating into the blood and make sure that the particle will get to its target. And to increase also the targeting and the way that those particles will be distributed into the body and make sure that it will come to the place where it is needed, we can use recognition moieties. So recognition moieties may be just antibodies or fragmented antibodies, but we can use also different type of ligands, such as the peptides or recognition for receptors. A couple of mandatory conditions, and these are mandatory for all type of particles. Basically, what we want them is to be degradable, make sure that they will be eliminated from the body. They need to be biocompatible, of course, not to injure the patients or to be toxic, non-immunogenic, so make sure that we will be able to treat again and again the patient with the same thing without having any adverse effects. And they also need to be physically stable into the blood, so to make sure that they will reach their target, which in this case is the brain. So actually, colloidal carrier do cross the blood-brain barrier. There are many reports in the literature. We had an example in the former presentation. I just picked up a few examples here from the literature. So examples from the group of Reuters, who has been one of the first to describe the passage of particles through the blood-brain barrier, and also the work from the people here in Modena. So basically using different type of polymer and different type of drug, it was possible to see that we get a better analgesic effect when the drug is presented as nanoparticles compared to the free drug. And also it was possible to see the better biodistribution of the drug, here the doxorubicin, when it is given as nanoparticles. Same thing by using derivatized PLGA and also derivatized PLGA with fluorescent marker, it was possible to see directly inside the visualization of those particles inside the brain, and also a better analgesic activity. So they do cross the blood-brain barrier. However, there are still a lot of questions that remains to be answered. And basically, what is the influence of the surface properties? I've just shown you that it is possible to change the polymer. It's possible also to modify the quality of the surface. However, it's very difficult to assess this, and it will be time-consuming to do it in vivo. We also need to evaluate the toxicity of the BBB on the BBB, because it has been asked several questions. We can maybe open the BBB, but we have to make sure that it closes afterwards. Otherwise, we may face a lot more problem than the pathology of the patient. Another point that is very important, and I think we have no real answer at the point, is how those particles get through the endothelial cells. So by using an in vitro model, that will be part of the answers. And finally, what we want to do also is to compare the drug permeabilities, whether they are associated or not with a carrier. So for this, the best way to assess that is to use in vitro models. There are several types of in vitro models for the blood-brain barrier, and at first they were developed just to understand how it works, basically. What are the connections between the different cell types and which factor is important and which is not. And we'll see, not very in detail, but I will give you a hint about the usability of these models for drug delivery system here. And so besides learning more about the physiology of the blood-brain barrier, these models have also and are still used to screen new molecules to see whether they would be able to cross the barrier. And the first one that was used is isolated brain capillaries. So these were developed in the 70s, and basically it's to take parts of the vessels from either animal or human samples. Of course, this brought a lot of information regarding how the blood-brain barrier works. However, it's a very delicate and very tricky method. It's very difficult. You don't have a lot of access to the material, and definitely it's very difficult to use it for screening purposes because it's a very fine material. You cannot have a high throughput screening method with this type of material. Some people have used also cell line from non-cerebral origin, and mainly because they would express some of the features of the blood-brain barrier, such as the tight junction, some enzymes, the presence of transporters, and also the efflux pump. Well, basically, this type of models are not very much to be recommended because they are still very far away from the blood-brain barrier properties, and you will not have all the features that will be expressed by this type of cells. So just to name a few, You have MDCK cell or KCO2 cells or ECB304 that I use. The advantage of these cells actually that they are very easily available in cell culture catalog, for instance. They are easy to grow and they are very well characterized. But again, they are very far away from what we want to see. The other model is to use primary or low passage brain capillary cell culture. Basically, the idea here is to remove the vessels from the brain and isolate the endothelial cells to have them in culture in vitro. So this is quite a nice model because you will have the cells with the real phenotype, and they will actually express all the markers, and they will have all the biochemical similarities with the blood-brain barrier cells. However, when you just take those cells and you put them in culture, very rapidly they will lose the typical feature when they are cultured alone. So basically, you will have a loosening of the tight junction. They will not express the different markers, the receptors, etc. So it has been shown quite soon that it was very important. What was very important was the environment of those cells and the fact that they were growing with other cells in vivo was also important in vitro. So people have looked at the influence of the basement membrane proteins and finally set up a few models and the proteins that were good. And to tighten even better is people have been working also on the medium in which you culture your cells by using more specific culture medium, by adding CAMP, vasoactive peptides, agonists, adrenergic agonists, or different type of factors. And finally, one of the most evolved here are also to use and to culture these endothelial cells with a medium that has been conditioned by astrocytes. So astrocytes are the cells that are in very close vicinity of the endothelial cells. But still with this, it's not exactly the same model. So the idea was also to co-culture these cells with the cells that they are in very close vicinity to in vivo. So people have been using glial cells, astrocytes, and also immortalized cells, so such as the glioma cells. And atoms have been done also by using neurons or pericytes. But basically, what gave the best results was to use with the glial cells. Neurons and pericytes are quite tricky to culture, and also there are very controversial data about whether they may influence positively or negatively the expression of the blood-brain barrier markers. So another way to have a model of the blood-brain barrier is to have immortalized brain endothelial cells, because of course using primary cells it's very time-consuming, because you have regularly to get new cells. You have just a few passages that you can use those cells, and then they will lose their expression of these markers. So basically, people have been using also immortalized brain endothelial cell. So you take the brain cells, the endothelial cells, and you make transformation by transfection and transduction and make sure that they will be able to express the different factors. So you have here a few of these cells that are available. Let's say that now there are about 20 of those cells that are available, and you may find them quite easily. And basically, what we can say with these cells is that they are a lot easier to grow than the primary cell line. However, they will not be as tight and will not express all the markers as would be the primary cell line. So it will all depend on what you want to see. I'm definitely not saying that these are not a good model. It is definitely a good model, but a model will stay a model, and you need to know what is your model before using it. So there are different ways of performing the culture of these cells, and the most used here is the static model. And in the static model, you have an insert, basically the trans wells, that are coated, so it's a filter, that is coated with the proteins that will bring the attachment of the endothelial cells on the top of this filter. And here you have just the culture medium with the different factor that will bring the differentiation of the to the endothelial cells. In the second model here, you have the co-culture, so the glial cells are first cultured in the bottom of the well, and after a while, when they are mature, then the endothelial cells are added to the well. So this is one, and I would say this is the most frequent model that we can see. And another model is described. It's called the dynamic 3D model. And this one is much more complicated system. However, it will bring a new factor, which is the shear stress that is brought by the blood coming into contact of the endothelial cells. And so basically this is made with a hollow fiber, and in this hollow fiber, inside you will have in the lumen, you will have the endothelial cells that will be grown, and on the external part of this fiber, you will have the astrocytes that will be grown, and basically you will have pumps that will pump regularly the medium through the hollow fiber system. So there are advantages, of course, of this one, because according to the authors, it's a lot closer to the in vivo situation, and because of this circulatory shear flow. Another advantage is that you can use over and over the same model for at least a couple of weeks, 10 to 15 days, just making sure that you have a washout session in between. You can co-culture different types of cells, of course. However, the negative point here, it's a very, very tricky setting, and basically there are two teams that are available to develop this model in the world. So one is in the US, and the other one is in Vienna, in Austria. And to my knowledge, poor small knowledge, it's very, very difficult to set up such a model if it's not in the hands of these people. So these are the different types of models that are available for the modeling of the BBB. And of course, if we are looking into details for the drug delivery system that we want to screen, of course, our model needs to have the specific properties of the BBB, cellular markers, and tight junction. There is one more complexity here because we are dealing with carriers. So we want to make sure that there will be no interaction of the carriers with the setting itself, and also that the setting will not be a limitation for the drug release of our particle system. So what we have done is actually we applied the model described by Delie and co-workers using glial cells here that are grown, you see, for 3 weeks. So it takes 3 weeks for the cells to get mature in order to receive then the filter with the endothelial cells on the top. And then it's another 12 days of co-culture before being able to get to the screening tests. And of course, you have here all the characterization of these models that is very close. It has been described in a lot of papers, so I will not go through all these markers. And it has been shown also that there is a very close correlation between in vitro and in vivo transport values for a large number of drugs. So basically, when we use this model, and every time you have a new model at home, I would say that you need, first of all, to know a little better about this. And what we've done is the Lucifer yellow permeability test, just to make sure that we do have a barrier and that the tight junctions are functional here. And what you can see here is the influence of the passage number. So what you can see is we can use these cells up to passage 6, and as soon as they go a little older, you can see that the permeability increases. So there is just a very small time window here where we can use those cells. And what we've looked at is also the effect on the time of co-culture with the glial cells. And what you can see here, so up to day 12, day 13, the permeability coefficient is lower than 1, and as soon as we go up and we go longer, day 14, then it goes higher than 1. So this of an impaired permeability, we cannot use them. So the first thing was to see what were the cells that we were dealing with and how we can use them. After that, of course, we looked at the particles, and so we've met very basic raw particles by using the nanoprecipitation method, which is nice because usually you have small particles smaller than 300 nanometers with nice polydispersity as well. So we use two types of polymers, PLA, very classical, in which we encapsulated DIO, which is a lipophilic fluorescent marker, and we also use a specific polymer made by Luca Constantino here in Modena, in which you have the rhodamine that is directly linked to the polymer. So we've made the two sets of particles, and we've watched what were their behavior with the cells and with our models. So first of all, we would just wanted to see by confocal microscopy whether we would be able to see those particles, and indeed. So in this type of experiments, we had an incubation with the cells that was 1 hour. Then we wash out everything, and we looked at the cell 24 hours after incubation. And what you can see, whether these are particles with rhodamine or particles with DIO, you can see nicely punctuated and fluorescent dots here, showing that we do have the particles. And we've done a control experiments where we use the DIO without any nanoparticles, and what you can see here is that the DIO will go into the cell membranes, and you don't have the same punctuated fluorescence here in this picture. So we also wanted to test the toxicity. Would those particles affect the tightness of the junction? So we left the particles for 1 hour with the cell culture, and then we run a toxicity permeability assay with the Lucifer yellow. These are the negative control, and here you have the permeability index, and these are different concentration of particles that we have used. And what we can see, we looked at this permeability 1 hour, 24 hours, and 48 hours after the particles were removed from the culture. And in the negative control, you can see that 48 hours we have an impairment of the permeability because we have a significant increase in the Lucifer yellow permeability. So we should not look at this result. However, if we look 1 hour or 24 hours after the incubation with the particles, you can see that there is no change in the permeability. So those particles do not have any effect on the passive permeability of Lucifer yellow. So more interestingly, we were interested in knowing how much of these particles were internalized by the cells, and these experiments were done with nanoparticles linked with rhodamine, where we looked at the percentage of incubated nanoparticles that were still in the intracellular compartment after 1 hour and also 24 hours after the incubation. And what you can see is that 1 hour after incubation, we have quite a few particles that are still here. And you can see this on the slides. And on the slides, this is the red little dots that you want to see here. However, when we looked at the same number 24 hours after that, part of the particles had disappeared. So basically, those nanoparticles are integrated into the cells, but somehow they don't stay in the cell. So the question is whether they are taken up and transcytosed or whether they are effluxed out of the cells. So for this, we've made a permeation assay. So with a 48 hours incubation. And what we have looked at is the quantity of particles that were internalized or that were present either in the luminal, here in red, and the luminal is the blood compartment, or intracellularly, and also abluminal, which is the receptor compartment. And very interestingly here, you can see that the particles do actually cross the cells in our models, and there are more, 48 hours afterwards, there are more particles in the abluminal side than into the luminal side. There is indeed a crossing of those particles through the endothelial cells. At the present time, the question remains, what is the way that this particle will take to go through the blood-brain barrier? We don't know. And since we are interested in screening here, we also wanted to see what would happen with different type of carriers. And for this, we use liposomes. And you can see that these are very basic formulation of liposome. There is no optimization so far at the time. We just wanted to know whether it would be possible. The critical point here was to be able to get small liposome under 200 nanometers in order maybe to compare this to the nanoparticles and also to have a very small polydispersity as well. And what you can see, the first thing, of course, that we run a toxicity assay. And what you can see, there is no change in the permeability of luciferase yellow, whether as a control or with 1 milligram of liposome in our test. So there is no toxicity. And what we've done, we also looked here at the confocal microscopy to see what happens. And we can see here those liposomes that are seen into the cells. They look to be in the nucleus, but I wouldn't trust this much. So this is where we are. It's still as preliminary results that we do have, but we are quite confident that here we have a BBB model that may be suitable to be a screening test for the permeability of our nanocarriers. The good point also is that neither PLA, PLGA, or liposome particles are toxic to the BBB in terms of permeability, so we can go on with this type of experiments. They seem to penetrate and permeate through the BBB. Now we need to know more about the ways and how we can improve the rate of uptake of this. And the same conclusion would apply for the liposomes. At that point, I would like also to acknowledge my co-workers and people from the University of Geneva in our lab, Eric Dolker, and mainly Aude Garcel, who has done all the work regarding, who has set up the model in our lab and who has done all the experiments. And Luca Constantino also for providing us the polymer, and William Fockett, who was a real help in putting this model up. And I would like to make a little advertisement here, since Aude left the lab very recently, and she is now she has taken over a job in Montpellier. So we have a postdoc position available for someone very motivated, would like to follow up on this study. So someone who is maybe not an expert, but knows quite a lot about cell biology and, if possible, BBB biology. Thank you very much.
Thank you, Professor Delie, for the interesting presentation. The discussion is open. There are the questions.
Thank you very much for this presentation. You may have mentioned it, but what's the origin of your primary cells? Are these rat, mice, bovine?
These are bovine and rats glial cells.
Okay, so you tried to, or you implemented exactly the same model as Cecchini. Have you been comparing with rat or mice?
Not yet. Not yet. So this is part also. And what I want to comment on this is a model, and I think also we shouldn't spend too much time in working with models. And as soon as possible, we should go to the clinics or to in vivo models. And that was actually the next step, was to sort of confirm these very preliminary results and go to animal model as soon as possible. Yeah.
But there's always a compatibility problem when you address issues on an in vitro model, for example, from bovine, and then you switch to in vivo models, mice or rat. Yeah. Have you been trying the porcine endothelial cells? No. Okay.
The first question is, are you sure there is no cleavage of the fluorescent probe from the particle? Because
yes, I think it's quite clear with the punctuation that we can see, and we haven't done the experiment with the rhodamine, but if you put the rhodamine labeled particles to sit for like a release study, there is no rhodamine coming out of those particles. So for this, we are pretty sure that they are still attached to the particle.
Excuse me, you mean if you leave them alone, where?
In a medium.
Medium, yes, but in the cell is different because you have cleaving enzymes.
Sure, but there is...
And also, I cannot remember, you did not show overlaying of the two different probes. But the punctuation, there was punctuation red and
well, actually we used the two type of markers in separate experiments.
Yes, but not together.
Not
because
together.
This is the indication that if they stay together, this is another proof of the fact that they are still intact. Otherwise, if you can... Yeah. Because well, as I'm sure, there are a lot of cleaving enzymes within the cells. In 24 hours, it could be... Well, this was the first question. The second is, I have no experience of literature, but as I can remember, many people used PLGA, not many, but people used PLGA nanoparticle for delivery. But on the base of your results, this seems to be at least in, I don't remember, 24 hours, you have a big, let's say, huge amount of particles in the passing through your model. But this is not has nothing to do with the results that the people, as far as I know, has obtained in vivo. So there is not such a high rate of success using PLGA nanoparticles in vivo.
Sure.
So this model is suggesting that you have in your hand something which is working, but...
in the literature, there is no data regarding the crossing of the nanoparticles into the brain. Basically, what has been done is to see that an effect was enhanced when the drug was used as nanoparticles compared to the free drug. There were also pathological, anatomical evidence that the particles had crossed the blood-brain barrier. But to my knowledge, there is no biodistribution of the nanoparticles into the brains, and let's say like X% of the dose is into the brain. So it's very difficult to compare. Now we are working with a model. So it all depends on the question that you are asking. My question is, I have maybe 10, 20 formulations that I want to use possibly in vivo, and I would like to know which one of those formulations is the best. So whether there is exactly the same ratio that will go into the brain when given orally or when given systemically to the animal, maybe not so important, as long as I will be able to show that I will have the same ranking of my formulation in vitro and in vivo. I think this is mainly the idea of modeling a barrier, is to see what is the ranking of our formulation. Will this one be able to cross? And at the present time, I don't have the proof of what I'm talking about. So I know that the only thing we have in hands is what I've shown. So at the time, we haven't done any experiments to show which different formulation will be better. And that would be the further steps. So to make a ranking with this in vitro model and to see whether this ranking is available, is good into the in vivo situation.
Thanks for the presentation, first of all, and for the effort in trying to mimic the BBB barrier, because as for all the artificial system, in vitro system, the effort is, of course, huge to be representative, or, as you mentioned, in order to find a way to discriminate independently upon what the in vivo situation could be after that. I mean, it may be superimposable or different, but we know that the physiological situations are normally pretty different from the in vitro simplifications. Based on that, what the main target of your work? I mean, is it to amplify, I mean, to exaggerate the differences between different nanoparticles, such as to gain a rank order that may discriminate the worst and the best? Or is it do you consider the system already validated with nanoparticles that may pass or may not pass, and the capability of the system is still in place? Or what are you going to do in order to refine the barrier, the model, in order to be more discriminative? Or
yeah, first of all, I think that our primary goal was to discriminate between the different type of carriers that we have in hands. And we have very new polymeric and amphiphilic polymers that may go to make polymeric micelles. And we definitely would like to compare the different types of carriers that we have here between liposome, polymeric micelles, and nanoparticles. This is one point. And if we want to go further, and to have like those particles decorated with some sort of ligands, we may have to change the model, of course, because these are cells that may not express the right marker. So then, in that case, we will be refining the model that we are going to use.
Any other questions?
The last comment I should make, actually, we did not develop the model ourselves, okay? We took the model from Celial, right?
Okay. Thank you again, Prof.
Thank you.
Delie.